Good morning, viewer. Welcome to our first active viewers. Here's the breaking news. Yes, plastic eating bacteria, genetic engineering, and environmental impact. Discover how plastic eating bacteria were discovered and re engineered to help tackle the world's plastic problem. Plastic eating bacteria could help to one day tackle some of the 14 million tons of plastic that is uploaded into our oceans every year. Plastic pollution leads to severe impact on marine ecosystems and can affect human health. For example, once plastic enters the ocean, it can suffocate and entangle animals, according to the International Union for Conservation of Nature, that is called IUCN. Microplastics are also ingested by many marine species that are both preyed upon by other species and that we catch for food. Once ingested, microplastics can leach the toxic contaminants that have collected on their surface into the body of the organism that has consumed it, according to the IUCN. Those toxins can accumulate and transfer up the food chain from marine life into humans. Whenever we eat something that has been taken from the sea, on land, the majority of plastic ends up either building up in landfills or burnt into incinerators, which releases toxic fumes. Just 16% of all plastic produced is recycled to make new plastic, according to the journal. However, in 2016, Japanese scientists made a remarkable discovery that could help tackle the world's plastic problem, according to the journal Science. Scientists collected plastic bottles outside a recycling facility and discovered that a species of bacteria was eating it way through them. Normally, bacteria spend their time absorbing dead organic matter, but Idonella saccharina has developed a taste for a certain type of plastic called polyethylene terephthalate, that is PET. After analyzing the bacteria, the scientists found that it produced two digestive enzymes called hydrolyzing PET or PETase. When these enzymes interact with PET plastic, it breaks down the long molecular chains into shorter chains, that is called monomers, called tetraphthalic acid and ethyl glycol. These monomers are then broken down further to release energy for growth of the bacteria. Following the discovery of plastic eating bacteria, many genetic scientists have experimented with Idonella saccharina to improve its efficiency. One such research venture has been to genetically engineer bacteria that are more efficient at enzyme production, such as Escherichia coli, and turn them into PETase factories. Although the discovery offers hope in the fight against mounting plastic, scientists caution that we are still years away from widespread commercial use. Similarly, PETase only decomposes PET plastic. There are 6 other plastic types that we are still unable to degrade using enzymes. Researchers at the University of Portsmouth have re-engineered PETase to create an enzyme cocktail that they say can digest plastic up to 6 times faster than normal. The scientists combine PETase with another plastic eating enzyme called MHUTase to form one super enzyme, according to the journal Proceedings of the National Academy of Science of the United States of America. The combined PETase MHUTase enzyme was created with a synchrotron, a type of particle accelerator that uses X-rays 10 billion times brighter than the sun. According to the University of Portsmouth, it enabled researchers to see the individual atoms of each enzyme and draw their molecular blueprint. Scientists then stitched their DNA together to form a super enzyme. This enzyme can also break down polyethylene Paranoid, that is called PEF, a sugar-based bioplastic. Researchers at the University of Edinburgh have been using E. coli bacteria to convert plastic into vanillin, the primary component of vanilla bean extract, considering that the global demand of vanillin exceeded 40,000 tons in 2018 and 85% is made from chemicals taken from fossil fuels. Using plastic could be an eco-friendly alternative situation, as Life Science has previously reported. After degrading PET plastic into its basic monomers, researchers took the process one step further and converted one of those monomers, terephthalic acid, into vanillin through a series of chemical reactions. The resulting vanillin is believed to be fit for human consumption, though further investigation is needed. Beyond this report for Shakti TV USA.